Hi to all of you. In this video, we will be discussing the nucleic acids, particularly focusing on the purine metabolism, how purines are synthesized by two different pathways, Denovo and Salvage pathway, and I will be discussing in-depth Denovo pathway of the purine metabolism, and rest will be discussed in the next lectures to come. Before we begin with the purine metabolism directly, we should be able to discern what metabolism is. Metabolism is actually referred to as biochemical reactions or the processes which take place within the cell or an organism, and because of these processes, they are able to maintain their living state. In the panel, you will see a number of different kinds of metabolisms of different macromolecules which take place in the living cell or organism, making it able to withstand life and exist in nature. All of these metabolisms are of different molecules which intertwine each other for the life to exist. Metabolism is further classified into two different types. One simply is called as catabolism, which is also referred to as destructive metabolism. They involve all those processes in which the macromolecules are broken down into simpler ones, into smaller and simpler ones from the larger existence. While as the anabolism is the reverse of catabolism, it is the synthetic process. It's also called as constructive metabolism. It involves all those macromolecules right from their smallest subunit. So it goes from simple to complex and helps in the synthesis of the larger macromolecules like glycogen, fats for the storage. A simple introduction to the nucleotide structure which you should know about before we begin the metabolism process is that nucleotide structures are containing the three things: ribose, sugar, nitrogenous base, and a phosphate, and they can exist as nucleoside or nucleotide. And then they can also exist as the ribonucleotides or deoxyribonucleotides. Whole introduction is in this slide. However, if you don't know about these things, then you have to go through the lecture which I have cleared in the nucleic acid chemistry. Go through that before you begin the metabolism process of the nucleotides. Nucleic acid metabolism is important in the living structures. Why? Because this helps in the synthesis of nucleotides and its metabolism as well, and it is essential for the existence of cell, because these nucleotides actually make up our nucleic acids, that is DNA and RNA, which are necessary for the genetic information to be carried in heredity. Furthermore, they are also helpful in making some intermediates in carbohydrate, lipid, and protein metabolisms like coenzyme A, FAD, NAD, as well as the energy currency of the cell, that is ATP in itself, sometimes replaced by GTP as well. And sometimes they also help in regulation of the metabolism by inhibiting certain enzymes. So this is importance of the nucleic acid metabolism. Starting with the biosynthesis, which is the anabolic part of the nucleic acid metabolism, as you can see in the accompanying picture, nucleotide biosynthesis involves two different pathways. One is called as the de novo pathway, another is called as the salvage pathway. De novo pathway actually means to synthesize from new, to synthesize from the scratch, and it involves the synthesis from the metabolic precursors. While as the salvaging is, as the name suggests, it tries to recycle the free bases or nucleotides which are released during nucleic acid breakdown, and recycles them to synthesize again the nucleotides for the living cells. Synthesis of purine part of the nitrogen base or purine nucleotide starts with the metabolic precursors, which are certain amino acids, ribose 5-phosphate, carbon dioxide, and the ammonium, which is donated either by the amino acids or in solution form. And the purine ring, which has to be constructed during the biosynthesis, actually is constructed on the platform or a backbone, which is ribose 5-phosphate. Ribose 5-phosphate is donated by HMP shunt pathway. And this ribose 5-phosphate is directed towards the biosynthesis of the nucleotides, and here particularly for the purines as well, is by converting it into PRPP5 phosphoribosyl 1 pyrophosphate, which is the first product which is then directed to the synthesis of purines by synthesizing inosine monophosphate in the first phase of the biosynthetic process. Denovo purine biosynthesis involves three phases which are categorized as such. First is the activation of the ribose 5-phosphate, which is the first step of the purine biosynthesis. Then follows the steps, around 10 steps of synthesis of the inosine monophosphate, which is the second phase of the purine biosynthesis. In the third phase, this inosine monophosphate is converted to either adenosine monophosphate and or guanosine monophosphate, depending upon the availability and depending upon the need of the cell at that time. Total de novo purine biosynthesis involves 13 different steps. As you can see in the picture, the purine synthesis and then different metabolic precursors which donate a specific atom to the purine ring is shown. You will see glycine donates most of the chunk of the purine atoms. Around 1/3 of the purine atoms in the purine ring are derived from glycine, and then there is another amino acids as well as the tetrahydrofolate which serve as the donor as well. So during the purine biosynthesis, the utilization is as given here on the list. One glycine is utilized, ammonia from the glutamine is used, folate from the N-formyl tetrahydrofolate is used, carbon dioxide is used, ATPs are used, about 6 of them till the AMP synthesis, and then when there is a conversion, there is a further utilization of the ATP molecules, and we require certain ions for the activation of certain enzymes for conversion and to do these biosynthetic processes. Step 1 or phase first of the purine biosynthesis is the activation of ribose 5-phosphate. This ribose 5-phosphate, as I already told you, comes from the HMP shunt pathway. I'll be carrying a separate lecture on the HMP shunt. So when a ribose 5-phosphate is derived from it, it is activated by specific enzymes which are PRPP synthetase or kinases. What they do is that they transfer the pyrophosphate moiety to the ribose 5-phosphate and result in the formation of the PRPP, which is 5-phosphoribosyl 1 pyrophosphate. Here you will see the ATP is converted to the AMP and its pyrophosphate moiety is bound to the C1 of the ribose 5-phosphate, making it PRPP. The reaction consumes the energy equivalent of the two ATPs, because here ATP is converted to AMP, and it also requires the magnesium ion for its activation of the enzyme. However, the activator is sometimes also the soluble form of the phosphate, and when you don't have to do the purine synthesis, the cell causes the inhibition of this enzyme as well with the help of certain purine nucleosides, which are the end products of the purine metabolism, that is either ADP or GDP or GMP as well. After activation of the ribose 5-phosphate to PRPP, this PRPP is then subjected to an 11 different steps to synthesize the inosine monophosphate. Total from the ribose 5-phosphate to the synthesis of inosine monophosphate, there are 11 steps. First step is the activation, and the rest of the 10 steps are called as the IMP pathway. The IMP pathway hence has step 2 to step 11, which we will cover now. In the first step of the IMP pathway, which is now the second phase of the purine biosynthetic process, it involves the conversion of the PRPP into the 5-phosphoribosyl amine. And here the enzyme is the amidotransferase, which transfers the amino group from the glutamine to the C1 of the phosphoribosyl pyrophosphate. and it dislodges the pyrophosphate out and inserts the amino group. This amino group forms the atom 9 of the purine ring. So the subsequent construction of the purine ring is based on this atom from now on. In the second step, the GAR synthetase converts the amine phosphoribosylate amine into the glycinamide ribotide, and here the whole moiety of the glycine is introduced into the purine ring, which makes up the atom 4, 5 and 7 of the purine ring eventually. Furthermore, ATP is hydrolyzed to ADP, hence the energy equivalent to 1 ATP is utilized by the synthetase process in this second step. In the third step of the IMP pathway, the transformerase enzyme converts the ribotide into the glycinamide ribotide, called as formyl glycinamide ribotide. Here the formyl group from N10 formyl tetrahydrofolate is utilized and added to the already added glycine unit. So the another atom of the purine ring is here added, which makes the C8 of purine eventually. In the fourth step, the FGAR, which is formyl glycinamide ribotide, is then converted into FGAM, formyl glycinamide ribotide. The synthetase process here utilizes the glutamine and converts this glutamine to glutamate, utilizing its own amino group, and which is added here, as you can see in the picture. So the enzyme involved is synthetase, and 3 of the purine ring is introduced here. ATP is also utilized to convert it into ADP, as it is a synthetase process. In the fifth middle reaction of the IMP pathway, the first closure of the ring happens. The closure of the ring, as you can see in the picture, is the imidazole ring, which is closed here by the help of a synthetase enzyme, which requires magnesium and potassium ions for its function, as well as it utilizes the energy from ATP, converting it into ADP, hydrolyzing it, and this energy helps in the closure of the ring and form the first ring, which is imidazole ring of the purine. In the sixth reaction, a carboxylase enzyme introduces the carbon dioxide into the ring and makes another atom of the purine ring and converts this air into carboxyaminoimidazole ring. So here the C6 of the purine is introduced by the help of air carboxylase. So this is a carboxylation reaction. In the seventh step, the care is converted into an S care, which usually involves the addition of a aspartate unit to the added amino group. As you can see in the picture, the aspartate goes here, and also this is a synthetase reaction, so ATPs are utilized here by hydrolysis. So N8 of the purine ring is contributed by the aspartate in this reaction. In the eighth reaction, what happens is that this aspartate unit, which was just introduced, is cleaved to release fumarate, which then goes into a TCA cycle. This is the link between the purine synthesis and the TCA cycle by the lyasing of the whole SKR to AKR. And here the enzyme is adenylosuccinate lyase, which releases the fumarate from the whole SKR and converts it to the reduced form of AKR. And here the next step will follow, which is the step 9. The enzyme here is transformylase, again utilizing the N10 formyl THF. Here the formyl group is added again, as you can see in the picture here, and the final atom of the purine ring is introduced in this step. In the final step, what happens is the closure of the another ring. The closure of the ring results in the formation of a complete purine ring, and here this is named as inosine monophosphate, and hence the second phase and the 10th reaction closes the IMP pathway by synthesizing inosine monophosphate at the end. This inosine monophosphate will serve as the precursor for the synthesis of either adenosine monophosphate or guanine monophosphate, depending upon the availability and depending upon the need of the cell. In the current panel, you will see that number of different steps which we just went through in a summary form, and there are certain kinds of inhibitors which can inhibit the rate limiting step of this whole IMP pathway, which is the first reaction of converting the phosphoribosyl pyrophosphate into the 5-phosphoribosyl amine, and this can be inhibited by AMP and GMP, which actually are the end products of this pathway. So if there is a ample quantity of AMP and GMP, it will be able to inhibit this rate limiting step and preventing the conversion of PRPP to phosphoribosyl amine form. Furthermore, as there are two important reactions which use the N10 formyl tetrahydrofolate, here as you can see in this picture, these two reactions are very much sensitive to the PABA analogs or folic acid analogs, and they can easily be inhibited by introducing sulfonamides or PABAs and preventing the synthesis of the IMP further down. So in summary, the utilization during the IMP pathway is one glycine unit and an ammonia from the glutamine 2 times, ammonia from aspartate, formyl group from folate 2 times in 2 reactions, then carbon dioxide unit, and an energy equivalent to 6 ATPs have been utilized until the synthesis of IMP. And we also require magnesium, manganese, and the potassium ions for the IMP pathway to smoothly function. Then the third phase of the purine de novo biosynthesis is the conversion of IMP either to AMP or GMP, and it both the conversions, whether IMP has to convert into AMP or IMP has to convert to GMP, which is a separate two pathways. Both pathways require two different steps. In the AMP production from the IMP, it involves the utilization of another aspartate and a GTP moiety, while as for the production of the GMP, we require glutamine, NAD, and ATP for the production, as you can see in the accompanying picture. There are two different pathways. The synthetic pathway of AMP is on the left hand side, and on the right hand side there is a GMP pathway, and each of the pathway has two different steps. For the conversion of IMP to the AMP, there are two different steps, as I already told you. In the step first of the conversion, enzyme is synthetase, which converts the IMP to the adenylosuccinate. Adenylosuccinate is actually formed by the addition of an aspartate moiety to the purine ring of the IMP. When the adenyloid moiety is formed, the GTP is hydrolyzed to GDP since it's a synthetase activity. So synthetase is dependent upon the GTP here and not on ATP. In the second step, this adenylosuccinate is lysed by the adenylosuccinate lyase, which removes a moiety of fumarate similar to that of the IMP reaction which we went through in the IMP pathway. This is another link to the TCA cycle, and it manages whether we have to have energy in the cell to synthesize the purines or not. So the enzyme here for the conversion of adenylosuccinate to the AMP moiety is the adenylosuccinate lyase, and here fumarate is also formed, and we get the conversion of IMP to the AMP moiety in these two steps. For the conversion of IMP to the GMP, similarly, there are two different steps. In the first step, IMP is converted to the XMP, xanthosine monophosphate, with the help of an NAD dependent reaction, which is a dehydrogenase type of a reaction. The enzyme here is IMP dehydrogenase, which dehydrogenates IMP to xanthosine monophosphate. In the second step, this xanthosine monophosphate is converted to finally GMP, guanosine monophosphate, with the help of a donation by the glutamine, which donates the amino group. This amino group is added to the purine ring, and converting enzyme is GMP synthetase. Since it is a synthetase reaction, it's dependent on again ATP. So this is the difference between the conversion of IMP to GMP. Here ATP is used. In the AMP conversion, GTP was used. These were the essential aspects of the de novo synthesis of purines. We will be discussing salvage pathway of purine in the next video. If you like this video, do visit my Amazon page for the number of different kinds of books which I have published. You can use them and order them in both Kindle and paperback format. And do subscribe to my channel for further videos which I will upload in due time.